I would say that one of the main conclusions of the meeting is the challenge of really using such a large amount of data to extract relevant conclusions from the biological standpoint. So we actually have been meeting to compare the data being obtained by different groups in different places using different technologies and try to put all the data together to try to understand how the genome encodes the biology of the living beings. I think it has been very useful for us also, both for the ENCODE people, but also for the scientific community in Barcelona, to have the possibility of interacting for the people working in Barcelona to learn firsthand which are the progresses in the genomic sciences at this particular moment, and in particular the progress within the Genome project. We hope that this will foster future relationship between Catalan scientists and scientists both working on genomic areas all around the world.
Our task is to try and understand how the genome works. So the genome is a very, very, very big place. There's a lot of it going on and we don't really understand it. By using experiments that directly look at how proteins bind to the DNA, we're able to understand better how it works and what different parts of the genome are doing. Now, this is particularly true outside of protein coding genes. So we've known a lot about protein coding genes for a long time, but we haven't really known how they're switched on and off in different timings. And the new experiments that we've done in ENCODE show many other things happening. In particular, there's a lot of RNA and a lot more complexity to the system than we originally thought.
The pattern of activity of these elements between different cell types is retaining memory of the early embryonic development. So that in the early stage, when you start out as a little small cell and you divide into different kinds of cells, somehow the cells are retaining memory of these events, which is reflected in the pattern of the regulatory DNA. The second part of the talk looked at a technology which we have developed called digital genomic footprinting, which allows us to look at the regulatory DNA at a very, very high resolution to see where proteins are binding in the regulatory regions. This technology allows you to do is to look at where proteins are sitting on DNA, but to look at all of the proteins at once. And we know which protein is sitting, or we infer which protein is sitting at a particular position by the type of DNA sequence that it's sitting on. And so what I showed was that this information is very, very quantitative, and that we can effectively measure how tightly one of these sequences is being bound by the proteins. And then we showed that you can look at large numbers of the binding sites for the regulatory proteins, and that they are following patterns that are characteristic of different cell types, brain cells, liver cells, etc.
We now estimate that there is something in the order of almost 50,000 genic regions and almost 140,000 transcripts, which means that the average genic regions have multiple transcripts. And right now the median is about 8 transcripts per genic region, most of which do not code for proteins. Just consider protein coding transcripts. We find that not surprisingly, that they have addresses inside the cell. The cell is not just a bag of molecules. So certain protein coding transcripts you'll find mostly in the nucleus. Others you'll find mostly in the cytoplasm. And if they're in the nucleus, they're in specialized addresses. Some of them are associated with nucleoli, some of them are associated with chromatin, and some of them are associated in the matrix in which the nucleus is made up of. So overall, the compartmentalization of these transcripts turn out to be an essential characteristic of these RNA molecules. Because for those transcripts which are in very low copy number, if you consider them being in this bag, all right, then the likelihood of considering them functional, all right, is probably remote. Because you're thinking, what is one molecule in this whole cell going to do? But if I tell you that one molecule is always found associated with DNA on chromatin, then you begin to think, maybe that one place is where it works. And since it gets taken to that place, right, in order to operate, that's why you can get away with making such a small amount of it.
I talked about allele-specific chromatin. This is how chromatin changes between individuals, and not only between individuals, parents, we have a situation where we have two parents, each with a child, so 6 different individuals, and we can see that the differences in the parents are also replicated in the children. This shows that differences in chromatin and how the DNA is packaged is somehow being inherited from the parents to the child. It means that we could look at this now, these differences in chromatin, in the context of human disease or other things. This is really the starting point for that, to show that we can measure these things accurately enough to do further investigations.
We study how transcription factors bind to DNA in living cells, and so we use a variety of assays to look on a genome-wide scale where any particular transcription factor is occupying a site in a living cell. Then we also combine that with measurements on a genome-wide scale of the RNAs that are made, messenger RNAs that are made from the same genes in the same regions that the binding occurs. And then we also combine it with epigenetic changes, especially in our case we study DNA methylation and look at that on a genome-wide scale. So by doing these measurements throughout the entire genome for multiple transcription factors and different cell types and cell conditions, before and after hormone treatment, for instance, we can essentially follow the patterns of the events that occur to regulate gene expression at genes on a genome-wide scale. Now we're applying these not just to cell lines, but also to primary tissues from cancer versus non-cancer cells, for instance, and in some cases other diseases like brain diseases.